Hello, my name is Franco Oitia. I'm the CEO of Caspr Biotech, and we're powering the next generation of CRISPR diagnostics. For a moment, let me take you to Laguna Alta, a lake at an altitude of 4,000 meters in my home country, Argentina. This place is considered to be one of the most extreme environments in the world. And here, we discovered a new CRISPR-Cas12 enzyme. Its novelty is given by the difference it has in terms of percentage identity when compared to any other Cas, while still having the key catalytic amino acids which make it belong to the Cas12 family. We filed a provisional patent which includes not only this Cas, but also our own Cas9. You might know about CRISPR in terms of its gene editing application. At Caspr, we're using CRISPR for something different. Through our Cas enzyme, we will revolutionize precision diagnostics by going into the genetic code of any DNA-based target, doing this better than existing technologies, as identifying specific DNA sequences has been the evolutionary purpose of CRISPR in nature. This precise search functionality is complemented with the collateral activity our Cas has, making it perfect for near instantaneous readouts. Here, you can see its performance for DNA degradation over time, which is achieved in less than 10 minutes. Initial results show our Cas has performed at par with the benchmark, and we're in the process of fully characterizing this. Freedom to operate will come from a license currently being negotiated with the correct CRISPR institution. Now, let me walk you through how we harness this enzyme as the tool for next-generation diagnostics. Our Cas is bound to a guide RNA, which is complementary to the DNA sequence of the proposed target. Both components together generate a CRISPR-Cas complex. If the DNA corresponding to the target is present in the sample, the Cas will find it and cut it. After doing this, it will go crazy, cutting all DNA in a non-specific manner. We evidence this collateral cut activity by adding a reporter system, a fluorochrome, and a quencher. So, if the target pathogen is present in the sample, a fluorescent signal is triggered. We're taking this groundbreaking technology to build a device to diagnose it all within hospitals, providing results in less than 30 minutes with minimal sample preparation and an attomolar sensitivity. And this is why medical experts are very excited with what we're doing. Right now, they use mostly PCR-based solutions to diagnose. When comparing face-to-face our technology with qPCR, we're able to obtain a 20% increase in sensitivity, a 30% increase in robustness, and best of all, we achieved this for just 1/3 of the cost. The Caspr platform will unify all detections at a molecular level, being capable of diagnosing for infectious diseases and genetic mutations. And we're starting with antimicrobial resistance as our primary indication, commonly known as the superbugs. There are currently two main techniques for its diagnosis. The first is the culture, which takes 2 to 3 days for results to be available. Other options include automated systems, but these require a culture enrichment, making the process last more than 24 hours in total. We will enable the diagnosis of antimicrobial resistant genes in a direct-from-blood format, providing results in less than 1 hour, reducing the unnecessary use of wide-spectrum antibiotics and shortening the period of patient isolation. Within a value-based US healthcare system, less time in the hospital means more money for the hospital, and we estimate this impact in savings to exceed $1 billion. We have done validations for the main resistances, this being KPC, MDM, and OXA, detecting for the conserved and distinct sequence of each of these targets, with our readout result being fluorescence. The downward slope corresponds to proportional dilutions in target concentration. And as this is highly correlated with the total amount of fluorescence, not only can we detect, but we can also quantify the amount of target pathogen present in the sample. The technology is there. Our only next step is to integrate this into a device. And we already started doing so. While here in IndieBio, we developed a disposable chip which integrates CRISPR with microfluidics. Our device will sell to hospitals for $5,000. Our business model will be based on the cartridge. This we will sell for $40 to $60 each, with a gross margin of 50% through distributors and 70% through direct sales, while also taking into account the corresponding CPT codes for each of our indications. CRISPR is set to disrupt what is now a PCR-based molecular diagnostics market, estimated to be worth $25 billion, only taking into account point of care. Other startups, such as Mammoth and Sherlock, are also using CRISPR, but they're focused on other targets, hence using alternative formats. Our go-to-market strategy has made one of these pioneer companies a possible partner rather than a competitor. Our system is the ideal solution for hospitals, and we're seeing this interest being generated from an early stage. We're currently doing a research-based pilot and are receiving LOIs from various medical institutions. We really move fast, and this is the team that's making it happen. Federico and Carla are PhDs in CRISPR system, with a combined experience of more than 10 years with this technology. I am a second-time founder, with my previous experience being a successful startup acquisition. The rest of the team is made up by people dedicated to other core areas, such as bioinformatics, microbiology, and synthetic biology. Our advisors are experts in infectious diseases, diagnostics, engineering, and FDA regulatory pathway. We're currently raising a seed round to advance in the development of our device and our cartridge technology, to then seek approval as a 510(k) with a prior clinical validation. Our seed round for $3 million is now open. Join us in the adventure of transforming molecular diagnostics while generating one of the industry's most exciting CRISPR-Cas IP portfolios. Thank you.